Welcome to the lesson on Chapter 10 about antimicrobials. So what we're going to start with here is a discussion on antibiotics. And what you want to know for this slide is the difference between a narrow and a broad spectrum of antibiotics. So the chart basically lets you know what's going on. But when you have a broad spectrum antibiotic, something like tetracycline, it can affect many different types of bacteria. It's going to be affecting your gram-negative, your gram-positive, and then rickettsias and chlamydias are actually intracellular obligate parasites, so they're even trickier to kill sometimes than many other types of bacteria. But tetracycline would be a good start if, for example, a patient came to the hospital and they're sick and you're not sure what their diagnosis is yet. So you start them on this broad spectrum and then you do some tests. Once the tests come in and you find out, for example, that it is in fact a gram-positive bacteria, then you could switch over to a more narrow antibiotic such as penicillin. So why would you do that? There's kind of pros and cons of both situations. The broad spectrum is going to kill a wide variety of pathogenic bacteria, but the downside is it's also going to affect your microbiome. So when you hear about people having side effects to antibiotics, there's actually a bunch of different side effects we'll talk about, but one of the most common, especially for oral antibiotics, is going to be gastrointestinal upset. And part of the reason for that is that your normal bacteria that are living in your gut are also being affected by the drugs, and so they're dying as well, and it's causing some commotion down there, and then you're going to have diarrhea or vomiting and things like that. The other downside, of course, of using the broad is that not only are you killing the healthy bacteria, but eventually you could develop drug resistance. So if you kept treating everything with broad spectrum all the time, you're going to get a lot more antibiotic resistance. So once you have your diagnosis, it is better to switch over to this narrow spectrum, and you'll notice some of them are super narrow. So for example, over here on this side, isoniazid is going to be specifically used for mycobacteria, such as tuberculosis, and also for leprosy as well. Okay, so those are the basic categories, narrow and broad. So next we want to move on to specific ways in which antibiotics act on the bacteria. So what's going on? So there's many different ways in which it can act, basically 5 different ways. So there's the cell wall inhibitors over here, cell membrane, affecting the DNA or RNA synthesis, folic acid, which also has to do with DNA and RNA synthesis, and then finally over here, protein synthesis. So what do you need to know about this chart? Unfortunately, a lot. You need to know a lot of information. You need to know how these drugs are acting. You definitely need to know what categories different drugs belong in. So there'll be questions that might say something like, which of the following is not a cell wall inhibitor? And it would have penicillin and bacitracin and vancomycin, but then it would also have polymyxin, and then that one clearly doesn't belong because polymyxin is down here. It's the cell membrane. It acts on the cell membrane, not on the cell wall. So there's questions like that. There's also questions on their mode of action, like what's actually happening inside the drug. There's questions on what type of bacteria they're better for, or whether they're broad or narrow. So there's a lot of detail in this chapter. Definitely read it really carefully. And what I'm going to do is kind of go over the highlights. So let's start over here with our cell wall inhibitors. So what are they actually doing? What they're doing is it's actually kind of specific, they block cross-links from forming in the bacteria cell wall. And by blocking the cell wall production, the bacteria actually pops open because the cell wall helps keep its structure. So I've seen little videos of penicillin being placed on top of some bacteria, and they literally look like they're just popping open on the screen. So that's pretty cool. The 2 major categories here are the penicillins and the cephalosporins. So let's start a little bit with the penicillin. I actually have a lot of information about penicillin. It's really, really cool drug. It's one of the first ones that was ever discovered all the way back in 1928, so almost 100 years ago now. And it was an accidental discovery. It does come from a mold. So there's a number of different molds in that category that can produce this penicillin chemical, and it's a natural antibiotic. So there's pros and cons for that. So natural means it's pretty easy to create. You can just grow the mold and then remove the chemical from it. We do have synthetic versions of it as well. Basically any kind of drug that ends in the term "-cillin", so penicillin, methicillin, ampicillin, all of those are derivatives, and they all have a lot of stuff in common, which is that they have a beta-lactam ring, and I'm actually going to show you that in a little bit. So they have what's called a beta-lactam ring as part of its structure. What's their main action? Of course, it's the cell wall inhibition. And they've been around for a while. So the downside of the penicillins is that there's quite a lot of resistance that has developed against them. Some of it was natural resistance that already occurred in the wild because bacteria were exposed to the mold in the wild. And then the other, of course, is the overuse of antibiotics, both in hospitals and also, of course, in the farm industry. A lot of antibiotics are prescribed to farm animals, and then that medication causes resistance in the wild bacteria in those animals, which can then be transferred to humans. So what are these cell wall inhibitors for? They're really great against your gram-positive bacteria, not necessarily so good against gram-negative, mostly because the gram-negative have that extra membrane wrapped around their cell wall, so destroying their cell wall is a little bit harder. It's harder to get to. Another thing to think about for penicillin is that quite a few people are allergic to it. Okay, so allergies. So it's not necessarily a normal side effect of the drug. Some of the drugs have side effects in general anyway, but this is an allergic reaction to the drug, and if someone's allergic to penicillin, they're probably going to be allergic to the other similar chemical compounds, and you're going to need a different treatment method. So what about your cephalosporins? Cephalosporins also come from a mold, so that's pretty cool. And they also do have a beta-lactam ring as well. So their mode of action is quite similar. It's a cell wall inhibitor. Now, why do we care about that? We care because if these bacteria have a natural resistance, how do they naturally have a resistance? What they're forming is inside of them, they have something called beta-lactamase. And if you recall from our very first lecture, ACE means it's an enzyme, and it's an enzyme that it destroys the beta-lactam ring. So it's going to destroy the chemical that is supposed to be harming this bacteria. There's actually two different versions. There's the penicillinase, specifically against the penicillins, and then there's let me see if I can spell this right, cephalosporinase. So how do we combat that? Well, we have kind of a clever solution, which is that you can add another chemical to your drug called clavulanic acid. And I apologize if I'm pronouncing any of these wrong. Some of these drugs I'm not that familiar with, so the pronunciation is kind of hit or miss. But that's okay for right now. Just kind of get familiar with how the words look. So when you mix this this acid with the penicillin or the cephalosporin, what it does is pretty cool. Is it blocks this enzyme. So if the bacteria had a beta-lactamase, now that's blocked. Therefore, it is susceptible again to the penicillin or to the cephalosporin. So that's pretty neat. What other things are on this list that you should know? So vancomycin, this has now become sort of an alternative to penicillin because of the fact that so many things are resistant to penicillin. However, it has its own set of resistant bacteria. So if you hear about VRE, it's the vancomycin resistant enterococcus. So that's kind of a problem now. And then when we were talking about MRSA before, MRSA is methicillin-resistant Staphylococcus aureus. So methicillin is just a modification of the penicillin. It's a slightly different version. What about here, bacitracin? This is one you'll hear about a lot. It's found in a lot of ointments. So your over-the-counter ointments for burns, cuts, scrapes, your typical Neosporin stuff, that is bacitracin. And another cool thing about that one, so penicillin and cephalosporin both came from naturally occurring molds. Bacitracin actually comes from Bacillus subtilis, which is a type of bacteria that produces this chemical. And then what else on this list? Let's see, isoniazid is important for treating TB, so treating tuberculosis, specifically the active form, so when the cells are actively dividing. The thing with tuberculosis, you'll see when we get to it again in our disease unit, is that it has developed a lot of drug resistance. And so most of the time we no longer treat tuberculosis with one single drug. We're actually going to treat it with isoniazid as well as rifampin and some other ones, because there's quite a few versions of the bacteria now that are multi-drug resistant and even extremely multi-drug resistant, which makes it very, very, very difficult to kill. This one, on the other hand, is a narrow spectrum. If you recall, going back to our previous diagram. So there it is, isoniazid is a narrow spectrum, specifically for a mycobacterium. The other downside of it is you often have to take it for 3 to 12 months. So if you've ever been prescribed antibiotics, a lot of times you only have to take it for maybe a couple weeks, like 1, 2, 3 weeks, once or twice a day orally, that kind of thing. But with this one, to treat TB, it can be a really, really long time. It's very hard to kill that bacterium. And the longer the prescription, the more problems you have with compliance, right? So compliance becomes a problem. People stop taking their medication, or they run out of medication, or they run out of insurance or money to pay for the medication, and they stop taking it maybe a couple months into their treatment. They start to feel better, right? So they stop taking it, and then now whatever bacteria is still in them is more likely to be drug resistant and then overgrow and become contagious to other people. So that, I think, takes care of most of the cell wall inhibitors. I think the other term I wanted you to know about there is the idea of selective toxicity. The idea that when we are when we're trying to prescribe drugs, we want something that's going to harm the pathogen and not harm the host. So cell wall inhibitors are really great for that because bacteria, of course, have a cell wall while the human cell does not. However, you look at some of the other categories here, like cell membrane and DNA synthesis, yes, we can target those in the pathogen, but you're going to have problems in the human cell as well, so you're more likely to have side effects and so forth. So speaking of cell membrane, let's move on to this next category. Let's see, polymyxins. These guys also come from a species of bacillus. So they're a naturally occurring antibiotic. They're going to be better for gram-negative. Okay, and that's because we're now targeting cell membrane. So the cell wall inhibitors weren't great for gram-negative, but these ones will be. And what they do is they cause the membrane to become leaky. Then down here, this one is produced by a different type of bacteria called Streptomyces. So not Streptococcus, that's something else, but Streptomyces are responsible for producing a whole wide range of chemicals that are antibiotics, and this is just one of the many that they can produce. And that's really all I have to do. All I have for these two. The cell membrane category is not very large, as you can see. So let's move on to this DNA synthesis one. Rifampin, again, what you want to know about that one is that it's mostly used for TB, so to help kill the Mycobacterium tuberculosis. It's also used against leprosy. Leprosy is another bacterium that's a Mycobacterium. By the way, leprosy is not even called leprosy anymore. It's called Hansen's disease. So it's just kind of fun fact. Okay, how about these quinolones? So quinolones originated from a substance called quinine. which is actually naturally produced by a tree in South America. And what it was historically used for is malaria, anti-malaria medication. And malaria is not a bacteria, it's a protozoan. So we'll talk about antiprotozoans more later, but since it's here, I'm just going to bring it up. So malaria, treated traditionally by quinine, and then there's some synthetic versions, chloroquine and primaquine. These are used to treat both active malaria and also can be used as a prophylaxis. So there's another vocabulary term that's good for you to know. So prophylaxis, the idea of taking the medication preventatively. If you were going to travel to Africa, you could take the medication. I forget exactly how long you have to take it, but let's say you have to take it for a week before you travel, and then you have to take it while you're there, and then maybe take it for a couple of weeks when you return. And the idea is that if you did get bitten by a mosquito that was infected with the protozoan, it would not be able to reproduce inside of you because the drugs would kill it. So that's an interesting kind of prophylactic there. We do not have a vaccine for malaria, unfortunately. Okay, the last bit of fun fact for this slide is that the streptomyces that produced this antibiotic here are also responsible for the precursor to rifampin as well. So now let's move along to the other side. Oh, actually, nope. I want to talk about this slide real quickly. This is just showing you what a beta-lactam ring is. So here you have a bunch of things that end in -cillin, right? So these are all derivatives of penicillin, and they all have the beta-lactam ring right there. It's just part of their structure. And then this side here, the R group, is the part that changes. So again, the downside is if you have beta-lactamase, then the bacteria is going to be resistant to all of these different chemicals because it can destroy this side of the chemical. But other bacteria might develop resistance to only a few of them because it might only be able to destroy one part of the molecule and not the beta-lactam ring. Okay, so moving on to the other side of this chart, and by the way, this chart is available on Adverum for you, so you can print it out and annotate it just like I am. Hopefully you'll find that useful. So let's look over here at the folic acid side since that's kind of briefer. So folic acid is also known as the B9 vitamin, okay? And what it is it's used for nucleotide synthesis. Which means you need it to make DNA. Okay, so if you block this inside the bacteria, it won't be able to make more DNA for itself. However, little fun fact about folic acid, it's also necessary in humans as well. So you may have heard of taking this extra B9 vitamin or extra folic acid during pregnancy. And what we found is that women who take enough folic acid are able to prevent neural tube defects. So neural tube defects. But if they don't get enough of this in their diet, then the baby might be born with neural tube defects, which means that part of their spinal cord might be exposed. You get spina bifida and some other different problems there. So what's happening inside of the bacteria? There's a couple different examples here, but it blocks the metabolism of making this folic acid. And so who do we use it for? We can use it against Shigella, which is a pretty nasty gut bacteria that can cause dysentery, which is bloody diarrhea, so not a fun disease. The other thing that it is good against is UTIs, so urinary tract infections. And then this one down here is specifically used in AIDS patients against something called PCP pneumonia, which, if you recall, PCP pneumonia, pneumocystic Urovecchi is actually a fungus, not a bacteria. So that's kind of interesting there. And that takes care of this category. So then let's move on to the protein synthesis category, and there's quite a lot listed there. So some interesting things about protein synthesis is that the ribosome of the prokaryote is different than it is in the eukaryote. I think I mentioned that in class already. But in the prokaryote, you have something called, it's right here, you have the 50S subunit and the 30S subunit that produce the complete ribosome, versus in a eukaryote, you actually have what's called a 60S subunit and a 40S subunit. So don't ask me what the S stands for. It just has something to do with the size and how it's produced, but they're different, okay? So the fact that they're different is a good thing. It means that you would have a little bit more selective toxicity here than you would with the cell membrane or with the DNA synthesis. However, it's not foolproof, okay? It's not foolproof. And the reason for that is, if you remember a little bit about mitochondria, okay, so mitochondria, there's that whole theory of endosymbiosis of where they came from, and the idea is that the mitochondria came from prokaryotic cells. Okay, so they have DNA that is similar to bacteria, and they also have their own ribosomes that are similar to bacteria. They're prokaryotic ribosomes, which means the drugs will impact mitochondria of eukaryotic cells. So how do they act? I'm actually going to show you that on a different slide, and then I'll come back to this one. So there's several different modes of action. What this picture is you've got your large subunit here. That's the 50S subunit of the ribosome, and then you have your small subunit, the 30S subunit of the ribosome there, and it's showing you how it assembles itself. So if this looks super confusing to you and you have no idea what's going on, you probably want to go back and review transcription and translation. So when DNA is transcribed, it is created into a new molecule called mRNA, and then the mRNA is translated to make proteins. So how does this happen? The mRNA has to join with the large and small ribosomal subunit, and then these little guys right here, these are called tRNA molecules, and they bring over the amino acids, and then the amino acids assemble to form the protein. So that's kind of the big picture. So what's going on here? All the different ways in which we can block this process. So in step one, well, I shouldn't really call it step one. How about in option one? In option one, you stop the entire assembly process, okay? So the ribosome cannot attach to the mRNA, and therefore protein synthesis is stopped. Done. Okay. In option number two, they were able to assemble, but you can see right here it says the mRNA is misread, therefore the protein will not be cracked. And if you have a bunch of malformed proteins, then that bacteria is going to die. In option three, the peptide bond is blocked. So what does that mean? It means when you have amino acids, so you have one amino acid, you have another amino acid, the way they link together is called a peptide bond. And you're supposed to have a whole bunch of them, okay, to make your protein. They just keep linking and linking and linking, and that happens here at the ribosome. So this bond is supposed to be formed right here. The ribosome helps you to do that, so it blocks that from happening. In option four, the tRNA is blocked, so that's that little, looks like a three-leaf clover thing right there. It can't attach to the ribosome, therefore it cannot help make the protein. And then in option number five, the ribosome is prevented from translocating. What that means is that the ribosome is supposed to move along down this mRNA, kind of reading it like a long sentence, right? It's reading it like one word at a time. So it can't do that, therefore it stops the protein synthesis. So anyway, those are just the different ways that this can happen. Going back to our drawing here, now let's look at some of the specific examples. Okay, so erythromycin is going to be actually created again by the Streptomyces bacteria. So Streptomyces is responsible for that. It's responsible for this whole group right here, which is pretty cool, and it was responsible for the daptomycin and the rifampin, if you recall. So naturally occurring antibiotic that stops protein synthesis. Some other things here on the list, Synarcid is something that's often used in surgery, like post-surgery to stop infections, and it's really good against staph, especially the staph that's resistant to so many other antibiotics. So staph is basically resistant to penicillin at this point, and it's also resistant to the methicillin, like we said, and sometimes it's resistant to the vancomycin, it's resistant to all this different stuff. So here you go, you have Synarcid instead. Next, these groups are in general good for gram-negative. Okay, so it's another way to target our gram-negative bacteria when you can't fight them with the cell wall inhibitors. And then I definitely want you to know tetracycline. That's a really, really common one. So some stuff to know about it. It is a broad spectrum. It's the most broad spectrum that was on our list. So it's very broad, but some side effects. Okay, so it's known to stain teeth, and you can actually see some pictures if you just Google some images of what can happen to people who take tetracycline. You'll see this like really dark, it's not even residue, it's just something dark that happens with the teeth as a side effect. It also can make oral contraceptives ineffective. Okay, so if someone is on oral contraceptives and they take this medication, they need to use an alternative method as well, otherwise their medication might stop working and they could get pregnant. Then down here, let's see, this one is another one that we use for MRSA. Okay, so another one that we can use there. And I'm just checking my notes, but I think that covers everything I want to say about this diagram. Okay, so now let's move on to different category of drugs. So antibiotics, of course, the big thing with antibiotics is that they only work against bacteria, though there were a couple exceptions on our list where, for example, we had the anti-malaria medication, but they don't work against viruses. So what do we do for viruses? There's really three main ways that we're going to stop viruses. One is we stop it from entering the cell. Okay. Two, we stop it from being able to copy itself. So let's say it was able to enter your cell, but we stop it from being able to copy its DNA or synthesize its proteins. And then three, we stop it from exiting as a mature virus. So those are the three kind of areas that we target. And let's just use this chart to sort of go through it. Okay, so this one is for step one. What we're doing here is maybe we can block the receptor. So there's a receptor on the host cell. If we block that receptor, then the virus can't touch it and cannot be brought into the cell through endocytosis. We could also stop fusion, or we could stop uncoating, right? Which means that maybe it starts to be brought into the cell, but it won't release its capsid and its nucleic acid. Let's see what else. Okay, so some examples here. Tamiflu, you might have heard of before. Tamiflu is specifically against the influenza virus, and what it does is it blocks the entry of that virus so that it interferes with its entry into the host cell. Okay, so category two, stopping nucleic acid synthesis. There's a couple different ways that it does that. So acyclovir, you may have heard of this one before. I think you often hear about it used against the herpes virus. And when we say herpes virus, by the way, there's so many of them. So it's not just like oral herpes or genital herpes. Those are actually herpes simplex 1 and 2. But we also have herpes virus 3, which is actually the chickenpox virus. We have herpes virus 4, 5, 6, 7, and 8. So there's a whole bunch of them. We'll learn more about them in future lessons. So moving on, what are some other ones that you should be aware of? AZT is one that was one of the first drugs used to treat AIDS all the way back in the 1980s. And what that one does is it's a nucleotide analog. So what does that mean? It means that when the basically when the virus is trying to assemble its DNA strand, okay, so let's say it's trying to assemble this DNA strand, and it has, A, C, C, G, T, A, something like that. What it will do is it will put in this fake nucleotide that isn't a real nucleotide, and it looks similar enough that the molecule thinks it belongs, but then once it inserts itself, it blocks the rest of that molecule, so the rest of the DNA can't be built. So that's pretty cool. So that's what AZT does. Then moving on to inhibiting the viral release. So that's our last category here. These guys just, I mean, it's exactly what it sounds like. They won't be able to bud. So this is called budding, right? Budding is when it leaves the cell and it stops it from being able to do that. You should know that HIV is now often treated with a combination of drugs. Okay, so treated with a combo, which is also sometimes referred to as a drug cocktail. And the reason for that is because viruses are notorious for mutating and becoming drug resistant. They mutate even faster than bacteria do. So we need to make sure we target it from many different places at once. So we're going to target the budding and the entry and the DNA synthesis all at once, so that it's unlikely that it could mutate and become resistant to all three of those drugs, at the same time. That's very, very unlikely. Actually, going back to HIV for a second, I don't know if it says it here, but the other thing that it does is it has what's called reverse transcriptase inhibitors. So if you recall, HIV has a special enzyme called reverse transcriptase, which is used to transform to transform RNA back into DNA. So if you block that step, it can't become latent. It can't hide itself inside of your host DNA. So that's another place where we can try to attack it. So those were our antivirals. Let's talk a little bit about antifungals, and I don't want to talk about them too much, but one that you will definitely see is called amphotericin B. Okay, so it's an antifungal. The big problem with antifungals is that they are in fact eukaryotic cells. Okay, so that's a problem because most chemicals or toxins that would affect them would also affect our animal cells, and that sucks. We don't want that to happen. So amphotericin B does tend to have quite a few side effects in humans as well, but it is a way to target the fungi, and what it does is it affects the cell membrane of the fungus. And it's really good against some fungi like Candida. We've talked about Candida, histoplasma, which is the valley fever that I think you read about in the case study, and then Cryptococcus, all of which can cause either pneumonia or meningitis are the two most common things that they cause. Also, there's a bunch of different drugs that end in azole, like that. Those are usually antifungals as well. Then let's talk very briefly about the antihelminths. So what's going on with them? They are also eukaryotic cells, and even more specific, they are animal cells. So you can't just target their cells. Instead, we have to look at what's something that would kill the worm but not kill the host. And the two categories that we do is one, we stop the microtubules of the worm from being able to absorb glucose. So a lot of these worms are, if you recall, a lot of them are intestinal parasites, or at least part of their life cycle is in your intestines, and for that reason, if you stop them from being able to absorb glucose, they're going to starve to death. Okay, so they starve and then they're dead. The other category that we can do is to paralyze the worm. So the worm is attaching itself to your intestinal wall, sometimes with hooks or suckers or all sorts of things. But if you paralyze it, then it will be released and it will exit via your feces. Okay, so that's another way to get rid of them. Not so great, of course, if it's a really, really big worm, but for some of the smaller worms, you can do that. And then just so that you can recognize some of their names, albendazole, praziquantel, these are anti-helminth medications. antiprotozoan. Let me see if I have anything really that I want to say about that. Not too much, honestly, other than the fact that malaria, Giardia, Trichomonas, and Entamoeba are all protozoans, and therefore usually antibiotics aren't going to work against them, right? So they don't have a cell wall, they don't have the same kind of ribosome as a prokaryotic cell, therefore they need their own category of drugs to fight them, and one of the drugs is called that one. Okay, so those are some of our different categories. Next, let's talk about drug resistance. Okay, so drug resistance is of course a huge problem, and there's many different things that especially bacteria can do to form drug resistance. Sometimes they create new enzymes, okay, so new enzymes. So think of that beta-lactamase. Well, beta-lactamase is just one example, but there are other kinds of enzymes that could help break down the chemical, and therefore it's no longer toxic to the bacteria. Another thing that they can do is change their cell surface so that they cannot uptake the drug. So the drug usually needs some sort of receptor for its ability to enter the cell, and if there's a mutation that changes the shape of that receptor, then the chemical can no longer enter. Another really cool thing that they do is that they can create these pumps, and the pumps actively pump out toxins that enter their cell. So yes, the toxin got in, but whoops, it's been pumped right back out. And sometimes these pumps are quite broad in their scope, so they may have a mutated pump that can actually pump out 5, 6, 7 different toxins, and one of those toxins happens to be one of the antibiotics that we were trying to fight it with. Another thing it can do is it can change its ribosome. So if it changes the shape or structure of its ribosome, then the drugs that specifically target ribosomes won't work anymore. And then finally, it can change its metabolic pathways. Some of them are interesting. They can shut down, not permanently, but shut down temporarily until the environment changes. We've, of course, learned about endospores, which means that it can just hibernate for a while. So it'll become an endospore, wait things out, and then once that chemical is gone, it reactivates, and you actually haven't killed it. Okay, so the very last, I think this is the very last slide I have, is toxic reactions. So looking at various types of side effects. Skin abnormalities, you're going to get things, let me use a different color actually because I feel like orange was too hard to see, but rash is not uncommon. Actually, one of my students last semester told me they had like a throat problem. They went to the doctor and they were prescribed antibiotics, but then their test result came back and it was negative for strep, so they pretty much did not have a bacterial throat infection. It was probably viral, but they were taking these antibiotics and they ended up developing a rash. So one of the side effects was that they got this rash on their skin. We talked, of course, about allergies in general, okay, especially penicillin, and it can be really bad. You can even have what's called anaphylactic shock, okay, that develops, which is a very extreme form of an allergic reaction. Other things that can happen, you could actually affect your white blood cells. Okay, so that sucks because your white blood cells are what you're using to fight the infection. Tetracycline, we talked about how it has this discoloration in the tooth. Quite a few of them will affect your kidneys, so we'll talk about that in a little bit. Yeah, here you go, see, kidney damage again. So kidneys are quite delicate, and your kidney, here it is again, kidney function. Kidneys are supposed to help eliminate toxins from the body. So if you take a medication that affects kidney function, then it's kind of a double slam because now the medication you were taking cannot be eliminated from the body. Plus, all of the other normal metabolic wastes that you were supposed to excrete also cannot leave your body. So that's pretty dangerous, and you could get renal failure from that. The quinolones, it says headache, dizziness, tremors. I think there was another one I wrote somewhere. It's not on this list, but The fluoroquinolones are even known to cause seizures. Okay, so there's some nervous system side effects there. Nausea, vomiting, that's pretty common. That happens with a lot of various drugs. Like I said, it can even just be from the disturbance of the microbiome in the gut. And then anything else over here? So AZT of course is for HIV. It can cause things like anemia. So what happens, of course, if someone has a side effect to drug is the doctor is going to try to prescribe an alternative, see if there's a different category that might still have the same result without having the same chemical side effects. And I believe that is it. Anything else? Oh, actually, let me just add one more thing. Well, there's no space on this slide, so I'll put it here. But it's not so much a side effect, but more just like a consequence. Okay, is overuse of antibiotics can cause overgrowth of basically your normal fungi that live in your body. Okay, so people who take oral antibiotics, for example, often get candidiasis. So they'll get oral thrush, or they might get a yeast infection. Why? Because they've killed all the healthy natural bacteria, and so then this nasty little candida takes over. So that's kind of one of the consequences. You also hear of Clostridium difficile. So what's going on with that one? That one's an endospore forming bacteria, and it often does live in the GI tract. So you might have like a very few of them living in your GI tract, and then if you take a lot of antibiotics and you kill your normal microbiome, then these guys can survive that, and then they take over once it's over, once the medication's over. Anything else? Oh, okay, so on the previous slide you saw, of course, kidney damage is a big one, but the other one is liver. So liver damage, due to the fact that your liver is supposed to be breaking down toxins, okay? It's supposed to detoxify. So that's its actual job. And you're putting extra toxins into your system, even though it's medication, it's medicine, it is still poisonous. You really have to follow the dosage recommendations. So dosage is going to be important. People's metabolism is going to affect it. So things like not just body weight, but your own internal metabolism is going to affect how quickly you process a drug. So the same dosage for the same size person might not always be accurate. You really have to monitor them and just kind of see how quickly are they breaking down these molecules. So you might take things like urine samples to see how much of the drug they're excreting and have to alter the dosage based on that. If people are mixing it with other drugs like alcohol, you're going to have much worse effects because alcohol already has a huge negative effect on the liver. So if the person's trying to take a prescription medication and mix it with alcohol, that's going to be bad news. Anything else? You hear about antibiotics being expired, so you're not supposed to take antibiotics that are expired. They're not going to necessarily work to fight the pathogen, but maybe they still have some chemicals in there that are damaging to the liver. Even your over-the-counter prescriptions will say things like, take 1 to 2 pills every 4 to 6 hours, but no more than 12 pills in a 24-hour period. So why are they saying that? They're saying that because an overdose can absolutely cause liver damage, sometimes fatal liver damage. So got to be careful with that. And then anything else I wanted to mention here? I think if we go back to this slide, just where do the bacteria get their drug resistance? Don't forget that they can do a bunch of different things. They can mutate, okay, so they can just mutate their own DNA, and then they can also have what's called horizontal transfer. And if you recall, what was that? That was things like conjugation, which is kind of like bacteria sex, but not really, where they use that sex pilus to pull each other together, and then one of them will transmit the plasmid over to the other one, and the plasmid might have helpful genes on it, and the helpful genes could be antibiotic resistant genes. So conjugation. You also saw transformation, which was that maybe there was a bacteria that was resistant to the drug, but it has died and been destroyed, but then the DNA from that dead bacteria can be taken up into a live bacteria, and now that live bacteria has this gene that is resistant to something. And then the last one was the last one was we did transformation, transduction. Okay, so that one was when a bacteriophage was the one that was transporting the gene from one bacteria to another. So those are just all different ways in which the drug resistance can spread super quickly, and that's something we want to try to avoid. Okay, so I think that does wrap it up, and that's my lesson for today.